I just want to say that this work would not be possible without an amazing team of collaborators. One really great thing about working at Stanford is that they do have 7 schools, and there's not really much of a boundary between schools. It's very easy to walk back and forth between them. And how this work really got started is one of my collaborators who's listed here, Niaz Benet. He's the director of the clinical pathology lab at Stanford. He saw some of our work on nanophotonics, in fact, cathodoluminescence optical tomography. And he was like, hey, can you use this to map out bacteria? So he just saw a press release, happened to read our article, and made the connection that like, oh, maybe what we're doing could help out a problem that he was having in diagnosing bacterial bloodstream infections. So it was an interesting question. Turns out that particular approach was not going to be ideally suited for the questions that he had, but it led into an entirely new and exciting direction for my lab, and that's going to be the first part of my talk, so how we can reimagine conventional microbiology toolkits for pathogen and bacterial detection. And then in the second part of my talk, I'll share with you work that we started I guess a little bit before COVID, but we really applied it to kind of viral, like both antigen and antibody detection after COVID started. This has been done in collaboration with Scott Boyd, who is the director of the clinical virology lab. So I feel pretty exciting having like this all-star team of colleagues at Stanford, including the director of the pathology lab, the director of the virology lab, and then many other amazing colleagues spanning computer science, the VA for clinical studies, surgery, and also molecular and cellular physiology, and then a great team of students to make everything possible. Okay, so like I said, here's the quick outline. I'll aim to spend about 10 minutes on each, and then I'll tell you a little bit about how my day has been since trying to balance doing research on these and take care of our little guys. So first part of my talk is on culture-free pathogen identification, combining an optical spectroscopy, in this case Raman spectroscopy, with machine learning. And then in the second part of my talk, I'll discuss COVID assays. We're working on both antigen and antibody assays, but in this presentation, I'll just share with you some preliminary work we have on the antibody assays using optical nanoantennas. Okay, so on the bacterial detection side of things, what I find fascinating is that roughly half of the cells in our body are not human cells, they're bacterial cells. So there are roughly 79 trillion cells in our body. 39 trillion of those are bacterial cells. And we've all heard about the microbiome and its importance in fighting off infection, in regulating our metabolism. So an incredibly fascinating research area. But of course, if a new bacterial species or invader enters our system, it's not always met with open arms. And now we know that viral infections are everywhere in the news. Bacterial infections don't really make headlines, but for sure they still have a global impact. And these can be bacterial infections ranging from E. coli to salmonella to tuberculosis. And if you look in the United States alone, a bacterial infection that winds up getting into the bloodstream, that type of infection, so a BSI or bacterial bloodstream infection, can cause more deaths than breast cancer, colon cancer, prostate cancer, and HIV combined. So to me, this is a pretty alarming statistic for such a nominally healthy or wealthy country, that if you take many cancers combined and AIDS, bacterial bloodstream infections are still one of the biggest killers. And then if you look more broadly to developing countries, roughly about 30% of all deaths are due to bacterial infection. And I think the statistics are that roughly every second a child under 5 dies due to a bacterial infection. So these to me are very alarming statistics, and reason to maybe rethink, why it is that the statistics are so bad, and if there's something that scientists can do to try to improve these statistics. And the reason why the outcome is so bad with bacterial infections, and in particular with bacterial bloodstream infections, is that you're really trying to find the needle in a haystack when you're doing bacterial infection detection and identification. So if you go to the doctor and they suspect that you have a bacterial bloodstream infection, what they'll do is they'll collect blood samples, and a bacterial bloodstream infection has symptoms that are probably similar to flu, like maybe you had pneumonia beforehand and then you started feeling ill. Actually, a number of new moms often will have symptoms of bacterial bloodstream infections after they've just given birth. So if they suspect that you have a bacterial bloodstream infection, they will collect blood samples, roughly anywhere from 5 milliliters of blood up to 20 milliliters of blood. And there's about like 1 bacteria per milliliter. So there are billions of blood cells, red blood cells, white blood cells, but then there's only that 1 bacteria that is causing a full-out immune response. So to be able to find that 1 bacteria or see if it's even there in a patient sample, what they have to do is culture the bacteria. So they put the blood sample in these blood culture vials. And in the simplest version, probably the most popular version that's in all microbiology labs, they have a pad at the bottom that essentially is a pH meter. So they load all of these into an incubator, and then they wait for the pH meter or the pad at the bottom to change color or change absorption, which indicates that there have bacteria have been multiplying and essentially changing the pH of the solution. So that tells them that bacteria are present. They still then need to identify which bacteria they are, which they usually do using mass spectroscopy or other like molecular-based techniques in a microbiology lab. And then the final step is to screen for antibiotic susceptibility. So they've identified it, but then they don't know which antibiotics to use. So what they'll do is put antibiotic patches on these agar plates and then see where the bacteria have died. And it's actually been fun sheltering at home because I can do almost this whole experiment basically in the 90-degree Bay Area heat with my 3-year-old and 5-year-old, and we've grown all sorts of exciting bacteria and then been able to look at them under the microscope. So detection can take 24 hours. Actually, there are some bacteria which are even more slow growing, and they can take multiple weeks to be able to detect. Like tuberculosis takes a while. And then antibiotic susceptibility testing takes another 24 hours. So it's a process that's just naturally slow. So meanwhile, you're in the hospital and you're suffering from symptoms. And if you have a bacterial bloodstream infection that becomes or progresses, every hour that passes without suitable treatment can decrease your chances of survival by about 10%. So obviously a doctor is just going to prescribe broad spectrum antibiotics or antibiotics that are, based on empirical evidence to hopefully alleviate your symptoms and address the infection. But according to the CDC, roughly 50% of patients are unnecessarily treated with antibiotics or are treated with the wrong type or dose. And that's, of course, giving rise to antimicrobial resistance. Hey, boys, you want to say hi? Yeah, I'm giving a talk, boys, okay? Marcus and Hugo just wanted to come say hi. So how can we rethink this conventional approach? Well, what we're doing in our lab is using a combination of bioprinting, which essentially digitizes the sample into single cellular droplets. And then what we do is we take essentially an image where we look at each, the color of each pixel. And the color is going to indicate whether that pixel contains a red blood cell, a white blood cell, or a bacterial cell. And of course, like the color is rather hard to distinguish, but what we do is use a label-free spectroscopy called Raman spectroscopy, where the laser light comes in and gets shifted based upon its interaction with the particular biological sample. So the advantage of this approach is that it requires no sample-specific preparation or cell tagging, so you don't need to know what you're looking for ahead of time. And we can also make the scattering from each sample quite strong using various like nanophotonic tricks, so dielectric metasurfaces or non-binding plasmonic particles. Okay, so I mentioned that bacteria don't necessarily have a particular color to them. You can engineer bacteria to have distinct colors, for example, by adding in various like membrane dyes. And that of course can lead to agar art, which is really beautiful. But for the most part, bacteria are rather colorless, especially when you're thinking about the 31 or so species and strains that are most commonly found in bacterial bloodstream infections. So how do we distinguish the color of natural bacteria without having to do any tagging? Well, what we do is rely on a technique called Raman spectroscopy. So what happens is we take any bacteria, we illuminate it with a monochromatic light source, say a laser, and then the vibrations from the molecular structure of the membrane give rise to a wavelength shift. So usually that shift can be of higher energy or lower energy. Most photons are generally scattered with the same energy, but a fraction of them are scattered with lower energy or higher energy. We're going to be looking at the Stokes shifted photons, so the lower energy photons, and we look at a Raman shift. So that's usually on the order of a nanometer to a few, tens to hundreds of nanometers from the light source. So we're going to be looking at the Raman shift. And the idea is that each cell has a unique composition, so we can use the scattering spectra, this Raman scattering spectra, to uniquely identify any cell. Okay, so if we look at the 31 species and strains of bacteria that are most commonly seen in bacterial bloodstream infections, we can collect single cell Raman spectra. And you can see that there are some species where the Raman scattering spectra is really different from other species. So for example, bacterial pneumonia has an extremely different signature than like E. coli, which has a very different signature from like Staph aureus. But there are other species and strains that look really similar and indiscernible. So for example, if you look at these particular strains of pneumonia or staph, like all of these spectra, for example, in the light blue and purple look pretty similar. So by eye, you can't distinguish them. And also using conventional algorithms like a principal component analysis, it's also very hard to distinguish the bacteria. But I will mention it's important that we can easily distinguish bacteria from blood. So that's very easy to do. Like with this vision of having a wide field Raman scanner, we can easily say like, oh, ignore all of these pixels because they're just blood. But then once we know bacteria are present, what we want to do is be able to uniquely determine which bacterial species and strain we're looking at. So we turn to developing a convolutional neural network. Probably many of you have worked with these in the context of images. Here we just applied a 25 convolutional neural net to one-dimensional spectra. So we input a spectra that is 1,000 elements long, and what gets output is a series of predictive scores that tells us the likelihood that a particular spectra that was input corresponds to a particular bacterial species and strain. So what we did is we took 60,000 individual cells that we knew the identity of. We trained the neural network, and then we fed in a new single cell spectra and said, like, with what likelihood does the algorithm correctly predict the identity of that spectra that it's never seen before. So the accuracy of our network is captured in this confusion matrix where we know what new spectra we're showing the algorithm, and then we look along the horizontal axis to see what it actually predicted it to be. So you can see for these three strains of Staph aureus, the algorithm does quite well. It does confuse, for example, strain 3 of Staph aureus with MSSA, which is like a methicillin susceptible Staph aureus strain. So there's a little bit of a confusion that happens there. So we have a misclassification. Staph epidermis and Lugedenicis does quite well. So on a number of trials, these are averaged over a few hundred trials, always gets those spectra correct. So then we can look at the full confusion matrix, or the matrix that describes classification accuracy of spectra that the algorithm has not seen before and what it predicts it to be. And you can see when you average over all of the spectra, the accuracy is about 82%. A lot of the misclassifications are actually in an isogenic strain. So I'll get to that in a second, but one of the strains in here we've genetically engineered to be identical to other strains except for one genetic difference. So that's where most of our misclassifications are occurring, in some of these genetically engineered pathogens, where we wanted to see to what extent can we look at, you know, if a pathogen develops antibiotic resistance, can we sense that genetic mutation? But what I'll mention is that many of these bacterial species and strains are all treated with the first choice of antibiotic. So I've colored them in light blue, light purple, orange if they have the same antibiotic treatment. And you can see that once you've grouped them together by the top choice antibiotic, the classification accuracy for the treatment you would use for that patient is actually extremely high. And this is what matters. For example, when you first go into the hospital with a bacterial infection, a doctor should know very quickly what antibiotic to prescribe rather than just giving broad spectrum antibiotics. So we have an accuracy that is comparable or actually better than mass spec in terms of determining the correct first choice antibiotic. And then, like I said, a lot of the misclassifications came with these so-called isogenic pairs, which are the same bacteria, but with the resistance gene inserted. But overall, I'm actually pretty excited by this preliminary data showing that we can predict, you know, just a single genetic difference with roughly 80 to 85% accuracy. And I think this is something that can be improved with like new spectroscopic techniques and additional upgrades to the neural net. And then I'll just kind of wrap up this particular part on the machine learning to say that we can also apply this to clinical samples. So I showed you all of the training data on cell line samples from ATCC, but we can also do this on clinical samples from Stanford and the VA, and we also wind up with equally high treatment accuracies. So how do we move from single cell data to a diagnostic prototype? And the idea is to take a patient sample and then print it into these single cellular droplets, and then design a wide field imaging camera, basically a wide field hyperspectral Raman scanner, where each pixel gives us a SIRS spectra. And then we have a clinical report that tells us like if a bacteria was present, and if so, which species and strain it is. So here's a video of the acoustic printing technique that we've developed for patient samples to print routinely single cellular droplets. The advantage of this acoustic ejection technique is that it's nozzle free. We're just relying on sound waves to eject single cell droplets. We can eject picoliter scale volume, so suitable for single cellular encapsulation. And we can process tens of milliliters of fluid within just a few minutes, so less than 5 minutes for a 20 milliliter sample. And the nice thing is that even though the acoustic ejection does get single cellular droplets, the cells still survive the ejection. A caveat that we've only done this on yeast, E. coli, and staph, but we're testing it over the full range of bacterial samples, and when we print them on agar plates, we can identify where we want to print them, and then all of the colonies grow from those single cellular droplets. And you can also you know, print in any well-defined array. So in this case, here are single cellular droplets, in some cases 2 droplets, that were printed from a patient sample. So in this case, whole blood. So that's kind of what the printing scheme looks like to print the sample out. And then we're designing the hyperspectral Raman scanner. Kind of a key criteria is that we did not want this to be a huge tabletop setup. We wanted it to be a smaller, like portable handheld setup. So what we did is we took our Raman spectra, and I mentioned that there were 1,000 wavelengths that were put into our neural network. What we did is identify which of those 1,000 wavelengths were most important for feature detection. And here I'm showing you the ones that are most important. So here we only have 300 features, all color-coded by the importance in terms of the classification. So you can see there wind up being roughly about 10 spectral bands that are most important for getting high classification accuracy. And if we just use those spectral bands rather than the entire 1,000 long spectral window, we can still get a very high treatment accuracy. So that allows us to move away from the huge like Raman setup that typically is used for high resolution Raman spectroscopy, and instead create a more handheld setup that costs less than $500 and still allows for good spectral acquisition plus high classification accuracy. So what we're working on next is integrating this into a full system that combines the printer with the handheld hyperspectral Raman spectrometer, and then moving into you know, a clinical trial, so to speak, or at least looking at far more clinical samples than the 60 or so that we've looked at so far. So I said I'd only talk about 20 minutes, and that was 20 minutes, but I apologize. Our little guys came in between, and were, I don't know if you heard them in the background, like excited for snacks and wanted me to play with them. But I'll go through this next part pretty quickly. Just give me about like 8 more minutes. So I want to discuss how we're taking some of our nanoantennas and using them for COVID diagnostics. So a quick like overview of like immunology. Many of you have seen you know, the COVID virus everywhere on the news. It's roughly 100 nanometers across. It has a variety of these spike proteins on the surface, which are about 10 nanometers long. And when the virus enters your body, some of the antigens or some portions of the spike protein will leak off. And those antigens can latch on to a B cell receptor. So here, BCR. So your B cells are in your bone marrow, and when they sense the presence of an antigen or an invader, then basically your B cell class differentiates, and you wind up generating a number of B cell receptors that are these Y shapes. Those B cell receptors, once the B cell is class changed, are called antibodies. So this Y-shaped structure is an IgG, an immunoglobulin G antibody. It's about 12 nanometers long, 14 nanometers across. And when your body is mounting an immune response, roughly about 2,000 antibodies are produced each second. Those antibodies will then latch onto the antigen, so the spike protein. And once a virus is decorated with antibodies, macrophage cells will come and destroy the antibody decorated viruses. So that's kind of a quick introduction to how the immune system works. One thing I find like pretty fascinating is just how large these antibodies are, which actually makes our life as you know, engineers of optical sensors a little bit more straightforward. So why would you want to have an antibody test? Probably many of you know this intimately from reading the news, but you know, first of all, it provides evidence that someone's been exposed to a virus. It also provides evidence that someone may have immunity to a virus, but of course that's going to vary a lot depending upon the type of virus. And you could also identify blood products like convalescent plasma that might contain antibodies that could be used for various therapeutic purposes. Some limitations of antibody tests right now, they're not as good as PCR testing for the virus itself if you want to learn if someone is actively infected. In fact, with the existing antibody tests, they can't tell or it's not sensitive enough to detect antibodies until a week or so after you have expressed symptoms. So developing antibody assays that are like almost as good as PCR and can tell you, you know, even before you're showing symptoms, if you're mounting an immune response would be really important. And then also ensuring that you don't have false positive or negative results. For example, a non-specific immune response if you just have seasonal coronavirus. So I've been pairing up with Scott Boyd to develop an antibody assay, and his lab and other collaborators that he's worked with have identified an antigen that is very specific to the SARS-CoV-2 antibodies that would be generated. So the antigen that he's working with is the end of the spike protein. It's the RBD, receptor binding domain of the spike protein, which here was imaged with cryo-EM. And why would you want to use the spike RBD domain as an antigen? Well, the prime reason is that it's very specific to SARS-CoV. So when you are using this as your antigen to test for antibodies, you will not get false positives due to other seasonal coronavirus. And then another reason to use this particular RBD is that it's relatively easy to express in large quantities, so you can make mass-produced antibody assays. So what's generally used, at least with my collaborator, Scott Boyd, who's doing all of the Stanford antibody assays, is an ELISA assay. This dates roughly from the 1960s, late 1960s. And the way the assay works is that you have plastic plates, essentially microwell plates, that are coated with the spike receptor binding domain. Then what you do is you add in the patient plasma. And then what you're doing is looking for a change in the absorption of the solution, or in some cases, a change in the fluorescence of the solution. So they add the patient plasma, you have some binding of the antibodies, and then basically they'll send in another HRP. It's basically like a horseradish protein that changes the absorption of the solution at 450 nanometers and allows you to detect whether you have IgG present or IgM present, so two different antibodies. So it generally takes about 5 hours to run the assay on a patient, you know, one day including the sample preparation and the data analysis. So actually kind of a long time. Ideally, you'd do something where you had like a quicker test that a person could perform even themselves. And also, like, it's very hard to get information about like antigen antibody binding, which I think is a really interesting question. Like, to what extent does the binding affinity or binding strength of the antigen and antibody determine like how well a patient might do in responding to the virus? So what we're doing is developing optical nanoantennas that are able to concentrate light into an extremely small volume, so that way you can sensitively detect changes in the environment, for example, for antibody binding. And then the exciting aspect about these particular nanoantennas is that they can control the amplitude, phase, and polarization of light. So I saw that Andre Farron is on the line, and he's worked extensively with these sorts of metasurfaces to, for example, do imaging. So you can beam steer light, or you can create lenses that are ultra thin, in many cases less than a micron thick. And what we're doing is designing these extremely resonant, so high quality factor resonant metasurfaces that will shift their wavelength when an antigen or antibody attaches, and then based upon the intensity at a particular position on the detector, we can massively multiplex the antibody detection. Okay, so how do these work? Here's just a quick tutorial. Let's assume we have an ultra thin slab of silicon, say 100 nanometers. We have light coming in from the top, so the wave vector K is indicated here. And what you would see looking in transmission is a series of Fabry-Perot resonances, where the transmission goes roughly from, close to 0, or in this case about 20%, up to about unity, depending upon the particular wavelength of light and whether it winds up being resonant in the film. So if we look at a wavelength of 1.47, this is out here, there's not really much happening. We're not at a resonance. So it's pretty boring. But the interesting thing is that this thin silicon slab also has a waveguide mode that free space light can't couple to, but if you were to come in with light from the side, you would get this waveguide mode. So you can see that the wave vector is quite a bit larger than the free space wave vector. And how we can couple to that waveguide mode is by adding in a grating or a series of grooves. So once we've added in grooves that are very shallow, say just on the order of 10 nanometers, if they have a period of 650 nanometers, basically that allows us to couple to a mode with an energy of 1.47 microns. And that particular mode is called a guided mode resonance. So now we've taken free space light normally incident on our silicon slab, and we have allowed the light to couple to that waveguide mode, generating a resonance within the structure that strongly concentrates the field. So here you can see the field is amplified by about 130x compared to how it was without that guided mode resonance. So you can see that strong peak here in blue, and then I've zoomed in. So the quality factor of these modes can be extremely high in the thousands to tens of thousands, approaching the millions. We're working on fabrication approaches to get such high quality factors. But I think a cool thing about these high quality factor metasurfaces is that they have Q factors that start to approach what you get in the microtoroidal ring resonators, but you have the opportunity to do free space incoupling and outcoupling. So in terms of a sensor, you can just send light through a lens that has this high quality factor resonance, and then just look at the intensity on the output side to image what's happening within your lens. Okay, so like I said, we have the ability to couple free space light in and out, and then we can also tweak the width of the silicon bars to steer light in particular directions. And you have several of the experts in metasurfaces for beam steering on campus, but here this just shows you how if you have all bars that are the same width, light just comes straight through. If now you start varying the width, you can see the phase profile starts to shift. That was just individual simulations, but here if I do a full field simulation, you can see light comes in and then gets beam steered. So our idea was to take or combine the idea of these high quality factor resonances with the beam steering ability of metasurfaces, so high quality factor plus beam steering, in order to do massively multiplexed biosensing. So here I'll just show you one paper showing that we can do high quality factor beam steering where the electric field gets enhanced by 100x, and you can see very sharp dips in the transmission spectra that correspond to those high quality factor modes. Experimentally, we have quality factors in the thousands that still enable beam steering. Interestingly, you can also do like spectroscopy as well. So you don't need to have a spectrometer. You can just make this grating essentially your spectrometer, so that way you image the spectra. And then what we do for antibody detection is we design the metasurface in such a way where more of the light actually leaks out of the nano antenna. So that way when an antigen binds, it gives rise to a little bit of a shift, and then an antibody comes along. Here we've just modeled that 12 nanometer long antibody with a polarizability that's comparable to what you would have for a protein polarizability. And you can see the reflection of the metasurface changes from less than 20% to close to unity as you increase the antibody concentration. So the hope is that this will be much more sensitive and also more portable or handheld than existing ELISA tests. I'll also mention this work has been a ton of fun to work on since the COVID shelter-in-place order came, because it's so multidisciplinary. It's been a lot of fun to work with our friends over in virology. I also have a really amazing team of chemistry students who have worked on tricks to control the surface functionalization, so that way we can selectively attach the antigens to the surface. So the idea is to use a pegylated matrix, so that way we don't have any absorption of other proteins that might be in the surface. So we maximize specific absorption. We also have nitroacetic acid, which can chelate the histag motifs on the antigens to orient the recognition site. This is something that the ELISA assay doesn't have, so we can basically get oriented attachment of the antibody, so we can precisely control how they're binding to the surface and not have any like steric hindrance that prevents the antibodies from binding. So it's been a fun approach that combines kind of expertise in photonics with expertise in chemistry. And then since being at home, couldn't go into lab, but like I said, I can culture bacteria with my kiddos, and we can take our own viral samples. And actually now I am an approved essential worker at Stanford working on COVID, but this is kind of a fluid chamber that I 3D printed for handling the COVID samples. So I'll just show you kind of a side profile of it. So bought a 3D printer. We have our meta surface here in the center, and then liquid comes in from the side. And I'll just play you this quick video where we've tested it with DNA. Here I'm just showing you with a purple paint from my kids, but you can see the liquid comes in, functionalizes the surface, and then goes out. So we can do multiple flushing or fluid flushing tests, all in a small compact profile. And then in liquid, before we could work with actual COVID samples, we were working just with functionalizing it with 25 base pair DNA. So that is roughly 5 nanometer long DNA. And with an unoptimized sample. So before we could go in the clean room, we just used some of the previous samples we had. We were able to get a 1 nanometer shift with DNA functionalized on this, or adding in the DNA. So I'm excited that with our optimized samples, we should probably get roughly about 5 times this shift in a fluidic environment. And hopefully this gives rise to a more sensitive and portable ELISA assay. So still lots of work to be done there, but we just recently got access to the clean room again, so we can make those samples, and then we just got hold of the first COVID assays. So what is the promise of this particular approach? Well, of course, metasurfaces can be made over large area. You can also extend them to detect not just antibodies, but also antigens if you wanted to make a detector of the virus or the antigen directly. So I'll summarize there. Like I said, these are 2 relatively new projects from my lab, but it was fun to share with you what we're working on, and I certainly value any questions you have, any feedback you have, like since it's so new. Pretty much all questions and all ideas are really good ideas. And talked about Raman scattering and machine learning to sensitively detect bacteria, and then also discussed COVID assays. And I apologize that I did not update my Outlook slide. This was from an older talk, but I do have one version of the talk that also includes feeding worms nanoparticles to measure forces, which is kind of fun. But I want to make sure I thank all of my lab members for making this work possible. Also our funders, including the NIH, the Moore Foundation, the Gates Foundation, and then the Stanford Catalyst. Huge thanks to Stanford for investing in a collaborative team. We won a competitive grant a few years ago that allowed me to get more into bio research. And then before I kickstart kind of the Q&A session on what I've been doing, I just want to share some pictures of what it's been like to be home. I guess you already saw a few interruptions with my husband and kids walking in. Right before the shelter in place started, my department actually went out for a dinner. And I think we'll probably look back on this in 20 years and say we were either brilliant or insane to go out for a dinner right before a pandemic. But you can see this restaurant, which is normally very crowded, only had 1 other or 2 other couples in the background. But it was a lot of fun to get to spend time with colleagues before quarantining ourselves. But since quarantining, I've had a lot of fun with my little guys. We try to get outside every day and go on hikes. I call this strength training. Both my boys learned how to ride bikes without training wheels. Marcus has been very good, but Hugo, our 3-year-old, just recently learned how to ride a bike with no training wheels. So we've been getting out for longer rides. And then I've been trying to teach them some basic spectroscopy. So we got diffraction glasses, and they've loved just checking out all of the rainbows and learning about the colors. And then in my lab, we make a lot of nanocrystals, but my sons, Marcus and Hugo, really wanted to make crystals. So we grew salt crystals. We've also grown sugar crystals to make rock candy. And then meanwhile, my students have kept me amazingly entertained with like all the extra skills that they're learning, from, perfecting their sourdough recipes to brewing beer. And here are 2 of my students toasting each other after they got a paper accepted with their own homemade beer brews. So anyway, thanks so much for listening. And like I said, happy to take any questions. Right now, I can actually only see Julia, Tiffany, and Jennifer, but I'm happy to look at any of your videos if you just want to make this a conversation. And all questions are good questions. So whether it's about science or life or things I'm doing outside of antibody assays in my garage, I'd love to chat with you. So thanks so much.
Wow, well, that was incredible. So much information in such a short amount of time. I actually received quite a few questions throughout this talk. So how much longer are we lucky enough to have you?
I can stay until about 5:15.
Okay, well, I kind of need to finish a little bit earlier, so I will go through some of them. So some of the questions that we received throughout were just can you explain a little bit more about the Raman technique? So one of the questions that came in was, what's the source of the Raman peak at 1,000 inverse centimeters that seems to appear in all the spectra? So that came in relatively early. And then to follow up, sort of to build upon that question, is what determines the Raman signal? And can the spectrum determine susceptibility for antibiotics with different mechanisms?
Yep, those are both great questions. So a lot of the peaks that are common to the bacteria tend to be membrane proteins that are conserved between different bacterial species, or various like carboxylic acid groups that you'll just see routinely in many of the bacteria, although some bacteria don't have quite the same arrangement, which is why you may not see that particular peak. And then in terms of can you determine antibiotic susceptibility, like that's one thing that I think the isogenic studies pointed to and that we're working on right now. Like, can we determine which, like not just the first choice antibiotic, but which antibiotic you would want to use, and what the minimum inhibitory concentration is for a particular patient. So some of the data that we collected right before the shutdown was looking at 1,000 or so E. coli samples from various patients, all with different antibiotic susceptibility profiles. And we've been training a network that right now, with above 80% accuracy, can actually predict which antibiotic that patient would be susceptible to. So it's a much more complicated problem, probably a far more interesting one, but definitely requires not only a lot of training data, but also like being aware of like which features you want to look for the antibiotic susceptibility, which requires, of course, knowledge about how that antibiotic is actually working, right? Like, is it targeting the nucleus? Is it targeting the membrane? So that way you can look for, within the Raman spectra, where there might be certain peaks that you expect subtle changes that would indicate the susceptibility.
All right. I don't know how to tell if the answer was well received, but I'll just say thank you.
If not, I'm happy to take more questions or chat with anyone offline.
I was going to say, like, I think maybe the best, because I don't get any feedback. So what I'll do is I will, Jen, can you see the answers, the questions on your end?
Oh, let me see. Maybe I can.
Maybe you can. Yeah. Okay. Okay, so then in that case, let's move on to the next question, because I'm leaving them all there, so you can look at them later if you wanted to.
Yeah.
Okay. So the next question is, how do you prevent biofouling on the surface, or off the surface rather?
Yeah, that's a great question. So right now, we're just trying to do quick measurements. So we do everything in a liquid chamber, like in plasma. And we basically plate the bacteria and then do sub-second Raman acquisition. So everything is collected quickly enough where, at least to the best of our ability, we have not seen biofouling be a problem. This is probably something that we would have to think about if we wanted to massively produce this, and if it would take longer between putting the bacteria on the surface and measuring the bacteria to make sure that there were no changes in the sample before we did the imaging. But right now, at least, we're just trying to do a streamlined process where we collect the spectra in the plasma fluidic environment without any either biofouling or bacterial death. So we're looking at live bacteria.
All right. Thank you. Yeah, that's actually a really big, I guess this question can be as broad or as, as focused as you want to make it. Here's a more detailed question. In one of your slides, you have different resonance responses below each, different resonance responses. Below each is written lambda is 55 nanometers, 57, so the wavelength, different wavelengths. Are those corresponding to one device or to different devices?
Oh, yeah. Yep, that's a great question. So those were 4 different devices where we had just changed the pitch to show that we can get different high quality factor resonances, and we varied the period basically of the grating to couple to that guided mode resonance, and that gave rise to a 30 nanometer shift. The cool thing though, and I didn't show this data, is that you can have multiple high Q resonances within one metasurface. So we've actually designed metasurfaces that have up to 5 at this point dips in transmission just in one device. But the data I showed here were 4 different devices.
Okay, I think that's, okay, that looks good. Oh my goodness, there are a lot more that came in. How does, I see, there's some questions about the career and there's some questions that are more, more scientific. So I think, Jen, if you could comment on how does the Raman technique solve the needle in the haystack problem? So if you're trying to detect bacteria using Raman spectroscopy, how does that,
oh yeah,
how does that enable you to find this one particular?
Yeah. So I did skip over that slide. So basically we can get extremely high accuracy with even just one single cell spectra. So already that's good news, right? We can identify the type of bacteria with just one spectra. And then the Raman on its own doesn't solve the needle in a haystack problem. What does solve it though is the digitization of the sample. So you have about 1 colony forming unit or 1 bacteria per milliliter of blood. So if you're drawing, and it's typical for a patient to have 4 or 5 mil draws, so you have 20 mils of blood, which corresponds to about 20 bacteria. So what we can do is in our hyperspectral Raman scanner, we can quickly filter out, okay, we don't need to worry about these pixels because they're just the blood. And then we go and do a high resolution Raman spectral scan of each of those, anywhere from like 5 to 20 bacterial samples that you would see in a patient sample. So you don't need to wait until you have, usually it takes about 10 to the 5th bacteria to do identification by mass spec. So we don't need to wait for that culture step. We can work with just the few bacteria that you would have in a patient sample. Or by the time it's brought over to the lab, in a way that's already a pre-culturing step. So we can work with many fewer bacteria than you would need for mass spec.
Wonderful, thank you. I think that we have a few. Are you ready for some more non-scientific questions, or should we?
Yeah, I think that would be fun, especially since you need to run off, and I can always take science questions like offline later on.
That would be great. Okay, so one question is that I'm going to read a bunch to you because I think there's a common theme maybe, and then you can provide your perspective on on those. One question is, how does an established faculty like you consider a scientist who would like to return to academia from industry after an interdisciplinary experience shifting from applied physics to more of physics for neuroscience. So that's one. Another one is, how did you decide you wanted to go to graduate school for a PhD and continue in academia for a professorship? Did you consider other career paths? And I think there's one more. What is the hardest part of being a PI?
Oh,
So
wow.
I think that there is just general admiration. And if you could comment on some of that, I think it would be great.
Yeah, so great questions. In terms of the first one, which I think was, how do I consider a scientist who would like to return to academia from industry after an interdisciplinary experience? I mean, life is a journey, and I would consider on a case-by-case basis. I've had, met some outstanding PhD students in our program who were in industry for a while and realized that they absolutely wanted to learn more and dive deeper into a topic, and wanted to return to get their PhD. So we've had in our own department PhD students who have basically finished their master's, went to go work in industry, and then decided they wanted to come back and do a PhD. I'd say it's a bit more unusual, I think, at least at Stanford, to find people who have worked in industry and then decided they wanted to be professors. But there are a few people in my field who have done that successfully. And I think if an academic job is calling you and you want to be in a profession of lifelong learning and work with some incredibly talented, creative students, by all means, please consider it. And I don't think anyone's going to hold it against you. I think people will understand that industry has different, milestones and priorities than academia. And if you have a well-articulated reason for wanting to make that return, by all means, go for it.
Those are your rollerblades down
And then in terms of how I decided if I wanted to go to grad school, generally, I really enjoyed being a student. I loved being around young, creative people. And I think at the time I finished my undergrad degree, I thought there was still a lot I hadn't learned yet. But more importantly, I just really enjoyed, being around friendly, energetic young people. So I applied to PhD programs and was lucky enough to get into some, including Caltech's program, which was amazing. And I'll say, I feel super lucky to have gotten into Caltech. I actually thought the admissions committee made a mistake, but I decided after the first few weeks there that even if they made a mistake, I was just going to ask as many questions as humanly possible, because I just wanted to understand things. And I think I learned more my first year of grad school than all 4 years of undergrad combined, because I finally got over the fear of asking questions. And I felt like I finally understood things. And that foundation has really helped me in understanding the connectedness between different fields of science. So, I'd say try not to plan too far ahead, because you never know what the future holds. But I think in most people's cases, the future is even better than you can imagine. And I know it's, probably hard to listen to that, especially when so many of you are dealing with, very diverse and difficult situations right now. But just try to, live in the present, be the best you and the best scientist you can be right now. And undoubtedly, you'll have opportunities in the future that you couldn't have even predicted at this point. So just try to do your best work. And I'm happy to talk about different career paths. I think being a professor is one of the most fun ones, largely because you just get to work with incredibly smart people. My students are some of the most creative, devoted people I've worked with, as are my colleagues. And you also get the chance to, branch out a bit. In no other profession do they allow you to start a company while you are a full-time employee of another business. So I think that's a really huge perk of being a PI, having great students, and then also, being able to explore alternate career trajectories in parallel.
This is so inspiring. I don't know about you, but I want to be a professor all over again now.
Well, now that you are, yep.
Actually, I am, yeah. How about we end with this question? I think this is very relevant. Just, do you have any advice for graduate students during the pandemic? I mean, your pictures and your research seem to suggest that there's no pandemic happening, and you're having so much fun with your family. But for graduate students, I think maybe some of them are maybe having a spectrum of emotional and otherwise response. So what would be your advice for?
Oh my gosh, yeah, that is an outstanding question. And I just want to start by, giving my kudos and congratulations to all of the grad students out there. I know that this must be an exceedingly difficult time for you, and I'm sure many of you are doing your best possible job to get through it and not only survive it, but thrive through the pandemic. But I can only imagine how difficult it would be. As a graduate student, I wound up meeting some of my closest friends, and I really thrived on the collaborative nature and the social nature of our lab. And I can imagine it's very challenging to not have that. So in terms of, coping with the pandemic, I'd say, first of all, try to see the forest through the trees. Like, this is, the world kind of slowing down, and an opportunity for you to reflect on where you've been and where you want to go, and try to use this opportunity to learn some new skills. Like, I'm sure many of you have a bucket list of skills you wanted to learn at one point, whether it's, machine learning, 3D printing, like cooking, making beautiful Illustrator figures, like polishing your presenting skills. But try to, whether scientific or not, just use this as a chance to, round yourself out as a person and gain some new skills that you might not otherwise be able to get. And then, as much as this might slow down the course of your PhD or perhaps change some of your career plans, hopefully if you can see the silver lining of the pandemic, it can be an opportunity for you to have spent, the last 10 weeks and perhaps looking forward the next handful of weeks more, becoming the person you always wanted to be. Like, figure out what your priorities are and try to see how you can possibly address that. And in my own case, kind of like Julia said, I have wanted to start a company for a while, and I feel like the pandemic, encouraged me to buy a 3D printer. It's a cheap one, but at least I could do some prototyping and, get started on problems that were really exciting for me. So in pathogen and viral diagnostics, those are areas that we hadn't really explored, in as much detail before. And it's been a lot of fun just to play at home. Like, we ordered agar petri dishes off of Amazon, cultured bacteria from, milk bottles, train sets. And then it's been kind of fun to just like buy homemade off the shelf components and try to construct systems. That said, I realize I'm in such a fortunate position to have, a good support network at home and to have an income, and happy to brainstorm individual solutions depending upon what situations you're in. But biggest piece of advice, just try to see the silver lining in it and try to enjoy it. Like, this is probably one of the biggest times we all have just to slow down and reflect on where we're going. I also left grad school during a huge economic downturn. So I sympathize with some of you who are thinking about like job prospects in the future. Try to the best of your ability not to worry about that and focus on the present if you can.
I think that this is a terrific note to end. This is so inspiring, and I think many of us could really brush up on our presentation skills, and seeing yourself in Zoom all the time also makes you realize that other people really are looking at you. So I think in the first maybe month of the pandemic, everybody was just kind of wearing PJs, and I think we're all kind of starting to come back to the reality of being professional, even on Zoom. So as things are starting to reopen, I think we're all, as we are all looking forward to it, but it's very refreshing and inspiring to see so much positivism coming from our screens at all of us. So thank you so much for taking the time and for prioritizing all of us over your own family.
Oh my gosh, well, it was so much fun. Thank you for giving me this opportunity. Hopefully I can see many of you on campus at some point, maybe in the next year or so. But yeah, I'm happy to answer any questions or schedule Zoom chats if anyone wants to have a one-on-one discussion about either science or career or life. But yeah, Caltech is awesome. So I think many of you are dialing in anyway as Caltech community members, and it's one of the best places in the world to be. So try to enjoy it, and I hope I can see you all in person in the not too distant future. Thank you so much.
Thank you.
Bye.
Bye, everyone.